Today we are going to look at one mechanism of controlling gene expression at chromosomal level, and this mechanism is histone acetylation. So histones, these are proteins in nature, and acetylation, it is now the process of adding acetyl groups to histone. Deacetylation, it is opposite, that is removal of acetyl groups from the histone. So histones, they are proteins and they are positively charged, and DNA is negatively charged. So these opposite charges, positive and negative, makes them to bind together with higher affinity and to form a compact complex, that is a nucleosome. So when you add acetyl groups, you will alter the charge on histone molecule, converting it from positive to a neutral charge. So this one will affect the force of attraction between histone and DNA, which was there before positive and negative. Now it is neutral and negative, so it will affect the force of attraction, therefore resulting to loosening of nucleosomes. After nucleosomes have loosened, the gene will be exposed, or the DNA will be exposed for transcription to take place. So here you can see my diagram here. This one, the one on blue is the one in blue color, it is histone protein. Then the one which is wounded around, it is DNA. So you can see the first one here, the compact structure. This is because of the negative or the opposite changes, positive for histone protein and negative for nucleic acid, that is DNA, made this compact to this complex to be tightly packed together. But now when you add acetyl groups in histone tail here, you can see the histone tail. When you add acetyl group, the histones are going to be neutrally charged, or the charge will be converted from positive to neutral, and this will affect the binding affinity in between these nucleosomes. Therefore, you can see the gene has been exposed, or the DNA molecule has been exposed for transcription, because acetylation has resulted to loosening of these nucleosomes. So that is how acetylation of histones is going to affect the gene expression at this level. The acetylation, when you remove again the acetyl molecule, you are going to regain the charge of histone, that is positive, and in between positive and negative, you are going to get a strong binding affinity, and these nucleosomes are going to be tightly packed together, and the gene is not going to be exposed to transcription factors, and therefore this one affect the process of transcription. So in nutshell, acetylation enhances transcription to take place, while deacetylation inhibits transcription from taking place. And these are some of the mechanisms which have been used in epigenetics, to study epigenetics. Thank you. Until we meet next time, where I will be talking about DNA methylation. Don't forget to like, subscribe, and share.